So I'm going to start talking about now these issues with, or issues, they're all a lot of issues anyway, with using fossil data and serial sampled data for in these birth-death processes and in this whole sort of concept of joint inference of both the tree topology and divergence times and placement of fossils for these analyses. And then after a break, or hopefully before the break time, we'll be able to start this total evidence tutorial. And so that's on the website too. All right, so I talked a lot before about like the two components of doing these divergence time analyses, right? So we have a prior on this clock rates or the substitution rates and how they're distributed across the tree. And then we also have these priors that describe how branching times happen over, how speciation events happen over time, and also how the topology is shaped, or our distribution of tree times and topology. So I did also mention that there's this other issue where the sequence data themselves alone do not confer any information about absolute time in terms of millions of years or days or whatever, and that we need some other information actually to get an absolute time scale on our tree. So without any of this additional information that can be provided that can give us information about absolute time, then we can really only estimate the relative ages and relative substitution rates for our data. So there might actually be plenty of questions that are totally reasonable to ask in that context. So you may be interested in understanding whether or not this clade of malaria diversified before or after another clade of malaria. And if they're all in the same tree, then you don't need to have sort of divergence times for those. That would apply also if you say we're interested in birds or whatever. However, if you need to do some comparison to some kind of external thing like biogeography, if you're interested in understanding if the uprise of the Andes had driven diversification in your group of plants, then you would need to know when diversification in your group of plants actually happened in order to compare it to when the uprise of the Andes happened. And so to do that, we need these branch lengths in proportion to this absolute time, the same time scale on which the Andes occurred, right? So commonly, the most common data for this type of problem has been from the fossil record when we're talking on a macroevolutionary time scale. The other side of that is that we can take things like serial sampled data from infectious diseases, so from an outbreak or an epidemic, and use those to provide a time structure to our tree. And so this is where that type of information is coming from. Another source of data for the absolute time can come from biogeography. And so and I'll talk about this a little bit at the end just to sort of lead into the biogeography that might be covered tomorrow. And what people have often done is say, if you're studying fishes that might be two species are distributed on either side of the Isthmus of Panama, given that we have a good estimate of the timing of the closure of the Isthmus of Panama, and we also are making the strict assumption that it was the formation of the isthmus that caused speciation in these two sister lineages. And given that, then we can say, we know that speciation happened after this isthmus was closed. So that might be another time that we can impose in our tree that gives us evidence that there was something that was associated with absolute time and our phylogeny. So these are the types of things that we kind of need to have if we want to make any of these inferences in an absolute time scale. So if we go back to this example that I showed before, I had this tree where we're assuming this strict molecular clock, and we found this fossil that represents the exact most recent common ancestor of taxon B and taxon C. And so this allows us to date the rest of the tree, right? The only issue with this example, well, not the only one, the other issue is that it's assuming a strict molecular clock, but this other issue is that we will never have that kind of certainty from the fossil record. we will never have the certainty to say, like, you know, this fossil bear is the most recent common ancestor bear of, you know, the polar bear and the grizzly bear. So we won't have that because the fossil record itself comes with a great deal of uncertainty. Not just in uncertainty in how these are related, but also uncertainty in the time of the fossil or things like that. So in a Bayesian context, Bayesian inference actually lends itself kind of nicely to accounting for uncertainty. And usually we might be able to say, like, you know, this fossil, it falls within the crown group of B and C. And by crown, I mean it's descended from the most recent common ancestor of B or C. We might be able to say, like, it is a sister lineage to C. And given that, we know that it's descended from the most recent common ancestor of B and C, and then we can assume that, like, B, or this most recent common ancestor, cannot be younger than the fossil that's descended from it. And then, given that we have observed this fossil, we have some uncertainty in the age of the most recent common ancestor of the group relative to the appearance of that fossil. So in the tutorial that you just did, you applied this sort of type of a prior on the root of your tree, right? So we had this caniform data set where we have bears and an outgroup, which is it's a gray wolf. And so we have a canid fossil in that example. And because that canid fossil we knew belonged to this group, then we could say, oh, it can date the whole group. So let's say C stands for canid and B stands for bear. So then we can say that that fossil, because it's descended from the most recent common ancestor of bears and canids, then we can date that caniform node. Is that pretty clear to everyone? What we're saying is a fossil is observed and the node that is ancestral to that fossil can never be younger than its descendant, but then we have to construct something that defines what our uncertainty in that age difference of those two those two parameters is. But so then in the Bayesian framework, what's typically has been done is to use these calibration distributions. So this kind of brings us to thinking about what fossils are in the context of our tree. And so this is actually a nice diagram from a paper by Mike Benton and Phil Donahue, who are both paleontologists, who wanted to provide like a sort of intuition for molecular and neontologists, molecular biologists and neontologists applying fossils as calibrations to understand what they're actually doing. Like, so when you're looking for fossils in the paleo literature, you often have a difficult time interpreting, what is actually meant by the placement of this fossil. And so they also address some real issues that comes when the fossil is pretty much misinterpreted in terms of where it's placed. So you can have, it's really difficult to describe these with words because they have big A's and little a's and stuff, but I'll try my best. So, here in terms of terminology, they talk about things like a crown group or a crown clade. And so here we have in this crown clade, a crown clade is defined as all of the descendants of an ancestral node and that node. So it'd be this node here and all of the things that are descended from it. So if we had a crown fossil, which is represented by this square, which I think is a little a, then this is a crown fossil, and we can use this fossil, which is the descendant of the most recent common ancestor of crown clade A, to impose a time calibration on that node, where we can say then A, or the ancestral node A, cannot be younger than this square fossil. So this is actually, ideally how we should be applying these types of approaches. But the other issue is that we might have a fossil that it looks like this white square here at B. And because it might look a lot like the crown clade of B, we might think it is in the crown of B. But what this is a stem fossil. So it diverged before the most recent common ancestor of all the living taxa of B. And even though it may look similar, placing this as a calibration constraint on this node would be a problem. Because what you see here, if this is the true timing of these divergence times, then by using this as a calibration on this node, we would put 0 probability that this node could actually be its true age. So that we'd put a 0 probability on the true age of that node if we use this stem fossil as a calibration fossil. And so these are really important things conceptually. And so within, the paleo literature, we might have things where we would say that B, this fossil is in the total group of B, and it's not so we might not know if it, because these things are often difficult to infer and understand. We might not know if it's in the crown of B or if it's a stem fossil of B, but we know that it's in that total group. So in that case then, B can only act as a calibration on the next node down. And so this is where it becomes important to really truly get a sense of where the fossils, how the fossils are related to the organisms in your extant tree. Does that make sense? Do you have a question?
Why are some of them white?
So I think, I guess this is something to do with more details in the paper. So these are age indicative fossils. So I think I don't know, Alan, I guess maybe you might have some interpretation of that. I'm interpreting that as that the ages are coming from, typically the ages are coming from the stratigraphic range in which it's found. And so if it's black, it actually can inform a node. So both of these are given the same age because, yeah. So these are actually, can be indicative of the age of this node.
It looks like he has the white ones just because
Yeah.
they're not being used as calibration
Yeah.
points.
Yeah.
From the same map, right?
So this, if you had all of these, you wouldn't use any of these fossils. You'd actually only use one of those, but they're the same time, right? And so this is sort of gives you a sense of like, even though you have access to a lot of fossils, they're not useful when they're used as calibrations. So coming back to this concept of these as calibrations, what we have is when we observe a fossil given some time, we know that it will impose a minimum age on the age of a node. So if we find this fossil and we're certain that it falls within the crown group of all these red taxa, then we know that it can impose a minimum age on the age of the most recent common ancestor of the red taxa. And this is saying there's 0 probability that this node can be younger than this descendant fossil, but then we have to determine like what is the prior probability of this node in relation to this fossil. But the one thing, this is how we can interpret the fossil record in terms of giving us like, age constraints for nodes in our tree, but we can't ever really get these really good maximum bounds from the fossil record. And that's because, if we see a fossil, we might see some, sort of evidence in the fossil record of the non-existence of a group, but, and in many cases, that might be good enough to construct a maximum bound, but in others it might not be. The absence of something is not maybe strong enough for you to say there's absolute 0 probability that something can be older than your maximum bound. So the common practice in Bayesian phylogenetics for a long time has been to use these minimum age constraints to construct these calibration densities for internal nodes in a tree of extant taxa. And so what these are parametric distributions that are offset by the minimum age provided by the fossil record. So this is similar to what you did with the root age in the exercise. And so what we have here is just showing you 4 different types of calibration densities that can be applied to a node in the tree. And what what this is, you know, we might be able to say, oh, what describes my uncertainty in the age of this node relative to the fossil is more of an exponential distribution, because the shape of this and the parameterization of this, you know, is the best match to my prior uncertainty. And so the nice thing about some of these distributions is they don't require you to specify a maximum age. So because these types of distributions, so not the uniform, but the log normal, gamma, exponential, and others have, you know, non-zero probability out to infinity. So you're not ever having to say, like, there's absolutely no way that this node could be, you know, this old. Except all of these distributions do require you to pick which one you want, but also then specify the parameterization of these calibration densities. So this is where they become pretty tricky. And so what these calibration densities are doing, so here we have a gamma distribution or a log normal, and if we choose one of those, we want that distribution to describe this waiting time before the speciation event and the appearance of the oldest fossil recovered for that clade. So, I mean, I guess some of you can, you know, if you have a lot of understanding of especially the fossil record in your group, this might be something intuitive that you can do. But for most neontologists, like most of us in the room, I mean, I think there's like only 2 paleontologists here. But most of us actually have a really difficult time specifying this, and I think even paleontologists do. And so, but what we do see, because if you remember the fact that, you know, the sequence data alone don't have any information about this absolute time scale. So all of your information about the absolute time on your tree is coming from your specification, your choice of and parameterization of these calibration densities and how they interact together if you have multiple of them. So specifying the appropriate calibration densities is really difficult when you're doing this approach. So when I've taught in these workshops before, I often get asked by, you know, people applying these methods to their own data, what is my prior on the age of this node? And the one thing I can never do is tell anyone what their prior belief is. I can tell you what mine is. It's probably, you know, anywhere from the fossil to, you know, maybe the age of the earth, or maybe the age of life, or animals if it's an animal or whatever. But, you know, beyond that, I think the uncertainty has to, you know, be shaped by what your understanding of your group is. And, you know, not just that, but also, you know, how these things might interact. You know, and that also sort of comes to this issue in a lot of the other software for applying calibration densities. So in BEAST and MrBayes, the way that these are done is that we've already talked about this too, we already actually have a prior on this node, and that comes from the birth-death process. So the birth-death process specified as our tree prior has a process, it gives us a prior on that node. And so in BEAST and MrBayes, what's typically done for these calibration densities is that they apply a second prior to that node. So even if you're describing your uncertainty in this age of this fossil to be shaped like a gamma distribution, the induced prior, by having these two things acting on this node, is actually giving you a potentially really different prior. And, you know, if you have multiple calibrations throughout your tree, those will also interact with you know, in certain ways. And so the priors themselves become not so much priors, but like sort of pseudo data or pseudo priors, because they're driving the inference in such a way that you know, they don't give us really sound sort of statistical basis of describing our uncertainty, because our uncertainty is really coming from the variance that we put in those prior calibration densities. In RevBayes, specifying an internal node calibration density is quite different, because we actually treat the fossil as a likelihood, as an observed data, and but the construction of these is kind of tricky. And if you're interested in it, we do have a version of the tutorial that does it, and we can go over that too for anyone who's interested. But I will make the case that I don't actually think for fossils that this is the most appropriate approach for calibrating a tree. And that's because, we really think, in a lot of ways, these calibration densities are quite ad hoc, where we're sort of trying to, define a calibration density based on something that we don't fully understand. It doesn't have strong grounds in a biological process or anything like that. But also, we might have a lot of fossils for our group, and we would actually really like to account for the fact that those fossils themselves are part of the same diversification process that gave us the living taxa, right? So, given that we already are saying, like, this fossil is a descendant of a node in our tree, that means it was part of the diversification process that gave us everything else, right? So ultimately, we should be able to account for that in a mechanistic way that includes the way that the same diversification of all of the lineages in our tree. Further, we'd actually like to use all of the fossils that we, know exist for our group. So, in this example from the bears, you only applied this fossil as a calibration, but there, are several other bear fossils that we can look to for our analysis. And if we use any of these as calibration densities, we take all 12 of these fossils. Are there 12 fossils? Anyway, we take we take all of the fossils and reduce them down to only 4 calibration densities. So there's a lot more information that we're throwing out and just using it as to construct this prior on the tree. So, in the end, we'd actually prefer to use all of the data and not throw out, a good chunk of it. So, but because of the fossils being part of this singular diversification process, then we can actually combine them in the birth-death process. And that's probably pretty much most of what I'll talk about for the rest of today. So this will require us to define this birth-death process, not just for having a rate of speciation and extinction, but also to have a rate of fossil recovery. So this will account for the rates of fossilization and preservation across the tree, and maybe potentially the fossil sampling in the end. So this kind of brings us to this sort of issue where it becomes really difficult to integrate the fossil record with like molecular and neontological data. And since I've been kind of working on these types of problems, I've found it very tricky to delve into the paleontological literature and other things like that on my own. And I think one of the issues is really there has been like this long time where the fields of molecular biology and evolutionary biology on modern species has been quite separated from paleontology and the field of evolutionary paleontology. And this is the case in such a way where it's like not escaped some people who are writing history of science and philosophy of science things, where they have observed, and I'll just read this part, where there's been this increasing realization that the barriers between these subfields, so paleontology and neontology, must be overcome if a complete theory of evolution and systematics is to be forged. And that's because there's this incredible amount of information that paleontologists understand about the fossil record. And if neontologists, if molecular people doing molecular systematics and interested in macroevolution on extant taxa are using paleo data without truly understanding what they're doing or the sort of structure of the fossil record and other issues like that, then there's a lot of things that are going to go missed, and that will lead to probably significant biases in our estimates of things like divergence time and diversification. And so this is the paper that I actually found that quote from, and it's just sort of was like interesting to me that there's just been this history of these two fields who are very focused on evolution and broad scale systematics that haven't been fully integrated. And that what it comes to is that one of the things that I think is kind of forging like a sort of resynthesis of paleontology and neontology has really been the statistical methods that have been developed to integrate these sources of data. So this brings us to two probably seminal papers that are kind of working in that direction. And one is this approach that was described by Paul Lewis, where he described a substitution model, essentially, for discrete morphological characters. So where many people had morphological characters, say of fossils or living taxa, and they're building trees on them, and they were using primarily just parsimony. And that's because there wasn't a framework that was you could easily plug into your 6-state discrete character into using in, say, something like POP and estimating with maximum likelihood. And so what Paul did was essentially just describe a model that you can take your discrete characters that has any number of states and essentially estimate a tree from those characters using basically what's like a Jukes-Cantor model. So it's saying that you have an equal rate of substitution between any state from one to the other. And so this is probably the simplest assumption you can make for DNA, just like it's the simplest assumption you can make here, but it gives us a mechanism to plug these data into a statistical framework. The second approach is this paper by Fred Ronquist, Sorina Klomfsting, and their colleagues, where they described a really comprehensive approach to jointly estimating the tree topology and divergence times of both fossil and molecular data in a sort of integrative Bayesian framework that uses this model for the discrete morphological characters. So these are two approaches that I think kind of will give us ways in order to combine the data and combine the sort of reintegrate these two fields of paleontology and neontology. So to do this, this I'm showing you now is a modularized graphical model. So what I'm doing here is this is still a probabilistic graphical model, but it's just sort of simplifying everything in these kind of compartmental units. And a lot of this you're already really familiar with, since you've already done this in multiple tutorials. So here we have a graphical model describing the generation of our DNA data, and all of these components you're familiar with. And you could imagine just plugging into one of these, a GTR model or a Jukes-Cantor model here, or this is like among site rate variation, so your gamma distributed rate heterogeneity and or your proportion of invariant sites. And then here we have our branch rate model, and that's what you did before this morning, where this is your relaxed clock model. So you can imagine that you have all these sources of data, and we can link them through one sort of underlying time-calibrated species tree. And so with the morphological data, and this could be for both fossils and living taxa, then you have the same types of processes. You just have different models for them, and so that's why you have different components here. And both of these observed data are coming from and conditionally dependent on this time-calibrated species tree. And given that we have some time tree, we also have observations from the fossil record that are our fossil occurrence times. So these are our data components, and this is essentially the sort of integrative framework that we're dealing with when we're thinking about combining these data. That we're linking them together with one component of our holistic graphical model, and that's really the tree. And as people working in evolution, this time tree is probably one of the things we're most interested in anyway. And we think that it's kind of driving a lot of what's going on in the observable data in our group. So this is our framework for combining these data. Does this seem pretty clear to everybody? So this is the approach, essentially, that we'll take, even in RevBayes, for doing this joint total evidence analysis. So this brings us to this idea of describing the fossil occurrence times as components or observations of our diversification model. And so what we've done here is to describe this in such a way where we have a serial sampled birth-death process. So here's what we do, so this is really called what we're calling the fossilized birth-death process, and this will become a a component of what you'll apply in the tutorial today. And so this really comes from a paper paper published by Tanya Stadler in 2010, where she described the serial sampled birth-death process. And these are figures sort of modified from that. So in this process, we have a complete tree. So when I talked about the birth-death process, before we start that process at some time, and this is what we're calling the origin time. And so here, this is a phi. And that's what we're using as the symbol for that. So given that we have some starting time for this process, we're assuming that there's some waiting time till a speciation event, and there's also going to be sampling events and extinction events over the whole course of the diversification of this group. And we also sample some of the taxa in the tips. Actually, I think in this particular example, we sampled all of the living taxa. But what we actually have is the reconstructed tree in the very end, right? So we've sampled some proportion of taxa in the present. We've sampled some of the fossils over time. But there are many lineages and things like that that will go unobserved, and we just won't observe those. And the model itself, because we might have some rate of speciation, extinction, we might have estimated a time for the origin, all of these combined can give us a probability for this tree. And given that we might have these observations of a number of samples in various parts of the tree, then we can compute the likelihood of those observations, given the model parameters. And so this is really what we're interested in estimating the probability of, is this reconstructed tree, but it accounts for the fact that there are these unobserved lineages and unobserved speciation and extinction events that we can't have sampled and may never will. So this process here, without all the red dots in it, is just like the same process that you applied in the previous tutorial. So it's a constant, we can assume a constant rate birth-death process that would generate these things. But if we also overlay on that a process that allows us to sample back in time, then we get the fossilized birth-death process. Is that pretty clear? Okay, so then if we think about the graphical model for this, it's not that much different from our graphical model of the constant rate birth-death process. We essentially just have the introduction of a single extra parameter. So our constant rate birth-death process has this origin time that it starts at, and so that's the time of when we had a single lineage. And we might put some priors on that. We have the speciation rate and the extinction rate with some priors on that. We have our sampling probability. Remember the probability you'll sample an extant taxon. But then we have this other thing here, and this is like the fossilization rate or the recovery rate. It's the rate at which we sample lineages in the process back in time. So the rate at which we get all these red dots. So this is simply the only extra parameter. So if you think about when you implement this in the Rev language, then you would just have this additional parameter that you would put some prior on. So if we look at this, what we're essentially saying is, given that we have this complete birth-death process, and we sample some number of taxa in the present, and we have these fossil occurrences that we've extracted from the fossil record, what these occurrences are, they are observations of our diversification process back in time. So as observations, these are actually data. So if we think about in the context of our graphical model, we could think about these observations coming off the tree with a gray circle here, because we've seen them, and that these observations are conditioned on the tree, and that helps us turn this into a likelihood for those observations. So, and then we can compute the probability of these observations, or the likelihood of these observations, given that we have these three parameters, as well as the origin time. So in a MCMC framework, what we can do is, if we have, say, the occurrence time of a fossil that is represented here with the skull and crossbones, we can think about how this might represent an observation on our tree, because it might actually attach to the tree at some time. So here's would be where it speciated off the tree. So if it had a if we had a speciation event for this fossil at this time here, then it would attach to the tree either at potentially either one of these branches, right? The other thing is we could in our MCMC sample a time for that fossil that's pretty much exactly or is exactly the same time as its occurrence time. And in that case, it becomes what's called a sampled ancestor and can fall directly on the lineage of either of those in the tree. So as we're sampling over MCMC, we may, at any particular state in our MCMC, compute the probability of any of these realizations. And it'd be all conditional on the parameters of the model. And then under this fossilized birth-death process, we can also account for the fact that there are multiple fossils that might be descended from a given node or might be, fall within our tree. So if we have this second fossil that is, observed at this blue time, and it might speciate off the tree, it can then speciate off either of the lineages leading to extant taxa or off of that lineage leading to its fossil relative. And if we happen to sample a time where it represents a sampled ancestor, then it can be a sampled ancestor of any of the extant lineages or of the fossil that's actually younger than it. So we can then use MCMC to marginalize over all of these reconstructions, and if we have morphological data, we can then also use those data to inform the placement of these fossils and those speciation times. So this is sort of the objective of using this approach in a Bayesian framework. And so this does bring me to this issue of the sampled ancestors. And I think one of the things is kind of the way that we think about trees, especially if we come from any school where we think about things in a cladistic sense, is that thinking about fossils as sampled ancestors is a little bit of a difficult thing to conceptualize. But this actually hasn't escaped the paleontologist. And so this is a paper by Mike Foote in 1996 where he talked about the probability of sampled ancestors in the fossil record. And that's because if you have a diversification process and you're sampling back in time, there has to be some non-zero probability of sampling a lineage that also has a sampled descendant. And so that's what a sampled ancestor is. It's a lineage that's been sampled, say a fossil or a virus or whatever, that also has a sampled descendant later on. And so if we look back to our FBD tree and our complete phylogeny, or the complete fossilized birth-death tree, here we have all the samples as the process of diversification is happening, we're sampling among the lineage that are existing, at a sampling event. And then we get to the end to our reconstructed tree, and we see that we have some lineages, or some samples actually several here, that fall directly on a lineage that also has a sampled descendant. And so because of these fossils, because our sampling is happening over this diversification time, there's this non-zero probability of sampling these sampled ancestors. If we use, say, some approach that doesn't allow sampled ancestors, then what happens is we end up inducing a lot of extra speciation events and extra extinction events in the tree that can then lead to some biases in our estimates of other parameters. So if you look back here, if this is our true sample, but then we have some birth-death process that doesn't allow sampled ancestors, what then that birth-death process is going to assume that there's extra speciation events here that didn't actually occur. And so this process is not very likely when we're thinking, in a macroevolutionary sense. There are some cases where we might actually see this type of a tree, and this really happens, I think, probably more with infectious disease. So if you are sampling HIV, typically when you sample a patient, they don't go on and transmit again because either they change their behavior, you put them on antiretroviral drugs, or they die. So in those cases, or cured. And in those cases, you might get something like this. Well, maybe these lineages would be a little bit longer. So then that's where you would have some other process directing the sampling of of those samples back in time. When we think on a macroevolutionary scale, there's nothing that says when this lineage fossilizes, it goes extinct or can never be sampled again. So because of that, then there's going to be this non-zero probability. And so postdoc April Wright and I actually did some simulations on this where we were just looking at, what is the proportion of sampled ancestors in simulated data? And so here what we did is we varied the turnover. So if you remember that turnover is the extinction divided by speciation. And as we increase turnover to something that's very close to 1, and also on the x-axis of each graph is increasing the fossil sampling rate. And so if you see where we have a situation where turnover is really low, so that means that extinction is really low. And even for very, very low rates of sampling fossils, we have a very high proportion of sampled ancestors. So many of the fossils that we have in this case are going to be sampled ancestors. And that as we increase turnover, we're going to have fewer sampled ancestors. That's because we have more extinction. But even in the case of very high turnover, we still have a significant number of those fossils are sampled ancestors. This does, so for all of these simulations, we simulated and included every extant taxon in the simulation. So you can imagine in a situation, and so in some of the empirical data sets we've applied this to, so we had one paper where we looked at like all Hymenoptera. So we had sampled, I think, every family within Hymenoptera, and there were just like a few fossils, maybe 13 or so. And so in that case where we have very few fossils and very few samples in the present, then we also see really, really low signal for sampled ancestors. But even then, the probability is not 0. So accounting for the fact that this is how the sampling might work becomes really important when we're trying to understand diversification. And if we don't marginalize over the probability or over the possibility that a fossil might be a sampled ancestor, then we might be biasing our results in certain ways. And actually, when we looked at these simulations, so this is just for one tree that we simulated, and if we look at the divergence times of nodes in that tree, so here in these little violin plots, we have the black line represents the true value, and these the green represents or is the posterior distribution of the node age for that node, given that we allow for sampled ancestors, and the gray is where we don't allow sampled ancestors. So in all of these, when we don't allow sampled ancestors, when they are present in our sample, we strongly bias our age estimates to much older ages. So ignoring these types of things can lead to really biased parameter estimates. It also actually really can bias your estimates of diversification and turnover, or speciation and extinction as well. So these are important issues, I think, that it's pretty critical to consider. So the other thing to think about is what we actually get in a lot of cases with the fossil record is we don't actually get single point estimates of fossil ages. Either these are given as ranges, stratigraphic ranges, where there might be multiple occurrences of a fossil lineage through time, where we have the first and last occurrence of Canis lupus is over this amount of time. Alternatively, they could also be intervals that represent uncertainty. And so accounting for this is also important. And so in the tutorial, what we'll do is treat these intervals as representing our uncertainty in the age of a single fossil specimen. And we can use MCMC to marginalize over our uncertainty and account for every possible age within that interval window. And the way we do that is we construct this in such a way that we're actually treating the interval, the median of the interval, as an observed datum. And then we compute the likelihood of any tip age given that observation. So here we have our FBD tree, and so if you remember the FBD model, there's all the other extra stuff. And coming off that is we can create a deterministic node that represents the age of that fossil tip, or if it's a sampled ancestor, fossil sampled ancestor. We also then assume that we have this, this other component of this interval that represents this, the median of that interval. And then what happens is we can compute the likelihood of this interval given that we have this tip and these, the observed minimum and maximum. And so the way it happens is as we sample the tip age, it could go with equal probability anywhere between this interval. But if we sample a tip age that is outside of this interval, the likelihood of this part of the model will be 0, and therefore the likelihood of the whole move will be 0. So what this allows us to do is just have our MCMC wiggle about in this interval and account for uncertainty in the age there. So this is one way of approaching that. And so here we're treating the model, this part of the model as the likelihood, and then this interval as data, as an observation. This is sometimes very sort of, it's hard to explain and also hard to understand. So as you implement this in the tutorial, I think it becomes a little more solid. But the important thing to think about is that now our fossil occurrences are data, and in order to put them in this graphical model framework, we need to associate a likelihood with these observations. So then we get to the point now, so we have our whole integrative frameworks, but we also are considering the fact that we have this other extra kind of data. So, most of you now today are familiar with these substitution models that we apply to molecular data, but now we have to have a substitution model describing this morphological data. And so these data are, can be treated in a lot of the similar ways as we might do with molecular data, because we're just considering them to be discrete characters that have some assigned state. You can see, though, if we do include fossils, that we're obviously going to have some missing data. And so this is one important thing to think about when you're dealing with the fossil record, is that there's a lot of things that don't preserve, right? So obviously all of the soft tissue of turtles, they don't show up in our character matrices. And often, like, a significant amount of the hard tissue doesn't either. In some cases, like those bear fossils, are represented by a single tooth, right? So there's a lot of filtering that the fossil record does in our ability to actually observe these things back in time, which is kind of sad, I guess. Probably sad for paleontologists. So the way that we treat these is to use this approach that was described by Paul Lewis in 2001. And what he really did in this paper was just created like a really basic framework for incorporating morphological data of any number of states into a data set. And so this just shows that we might have some character that has 3 different states, so we are coding them with numbers, so from 0 to 2. And so then we just create a Q matrix that basically says you have the same equal rate of going from one state to another. And here, this is describing a generic one for K number of states. So you could imagine this going out to 9 states or more. And I think there's some tricky ways of putting or more than 10 states in your nexus matrix, but we're only going to be dealing with binary characters today. But then essentially what the sort of simplest version of this model is assuming that transitions between, say, state 0 to state 1 and are essentially symmetric in every case. So you have an equal rate of going from 0 to 2 or 0 to 1 and back and forth from all of those. So this is the simplest approach. It's essentially a Jukes-Cantor model. And so in our tutorial, what we'll do is apply this to a binary set of characters. So when we think about what are morphological characters that are binary, these are essentially presence or absence things, or how, like, is it blue or gray or something like that. There's only 2 states by which this character can take. So this is just one example, though, of a really simple model. And so if you're really interested in more complex models of morphological character change, there is another tutorial on that as well, and maybe that's something that we could discuss maybe at the end of the week on Friday or something too. And so in those cases, we might construct a model where we have a different rate of going from 0 to 1 versus 1 to 0. And that if we say have several like binary characters in our analysis, we might then also include among character rate variation for those as well. So you can think about a lot of the things that you might do for your molecular data, you can do for these data too. An important thing to think about though is that there are very critical things that are specific to the filtering of morphological character data. And that has to do with really how they're described. And so when we might code morphological characters, we're never going to include those characters that are invariant for our entire, or constant for our entire group of extant taxa. So if you were working on mammals, you wouldn't have a column in your matrix for hair or mammary glands. And so that, if you think about it, what our models are assuming is that we're actually drawing at random, like, say, DNA sites from the genome, that these are IID. So given that, we would have some sites in our molecular alignment that have all A's at some site. But our morphological character matrices don't tend to have those. They also don't tend to have characters that are not parsimony informative. So this would be one where you just had, if these were all, if this was a 1 and this was 0, this would be not a parsimony informative site. And so we have to account for these, this aspect of our data. And that's done in the tutorial too. We can include these corrections that account for the fact that we haven't observed all of the types of characters that might have evolved. And so for some of you who work on more molecular or genomic data, then there's also, it's also worthwhile thinking about these types of models so you can apply these types of things to things like gene content data. And so you might be interested in saying, looking at all of the orthologous genes in your entire genome and saying, is the presence or absence like something that you can treat as an evolutionary character? And in those cases, you also have this issue with filtering, right? So if you're looking at gene content data, you're not going to have represented in your matrix those sites, those genes for which were all lost in your group because you can't possibly observe them. So then there are ways that we can account for the type of data filtering. So that's a really important thing to think about. With morphological character data, there's also these other issues too, right? So we can say that, like, if this is, the presence or absence of, a third scute on your turtle shell, and then we have another character that represents, a certain shape of shell. But they're both going to be coded as 0 to 1. So if we use the same matrix on those two characters, what does it mean to say you have a rate of going of, 0.25 of going from having the scute to not having the scute to having the scute, and you have a rate of 0.25 of having, a square egg versus a round egg. And so if that is meaningful, maybe that makes sense but it doesn't, it's meaningful and makes sense if we apply those types of generalities to molecular data because biochemistry gives us some sense that going from an A to a C in one character in one locus is likely to have the same rate as going from an A to a C in the neighboring site within that locus. We're making those assumptions when we're applying a singular model to a set of molecular data. And it might actually make more sense, right? And here it makes a lot less sense. So these are important things to think about when you're applying these. But one thing that makes it really hard is we don't have strict general models for morphological character evolution. And so maybe there's some ways, I think, we can have, other ways of applying morphological data in these analyses. One of the things that I'm interested in is really, bringing in geometric morphometrics into this context as well, where you have, a slightly more objective but still highly subjective approach to collecting data, and maybe that you can identify homologies in certain ways. But it's still very difficult to bring these data into the morphological character data. Does anyone have questions on those? So when we have all of these model components together, so I think I've talked about all of them now, we can then now link them together and do our full total evidence analysis. And so what we're thinking about now is what our data are these time intervals and then observations of living taxa, and we might have things like characters associated with them. And so our objective of the total evidence analysis is to reconstruct the whole diversification process, and allows us to this will allow us to understand what the diversification process was that gave us our sampled fossils, our extant taxa, as well as all of these dynamics that were driving that process. And so ultimately what we're attempting to estimate and account for is this reconstructed tree. So I was just going to talk about an example of an analysis that we've done recently. And so this was a study that was in collaboration with Dan Sepka as well as several others. And we have these now approaches for doing this joint analysis of divergence times and phylogeny and accounting for these this diversification process that gave us both the fossil and the living taxa. And we were interested in determining if we applied these to a group of organisms, particularly these penguins, how does that change our understanding of the diversification of penguins in deep time? And so Dan is actually a penguin paleontologist and has a really amazing understanding of the diversity of this group. And so if we're interested in penguins like Dan is, and like probably everybody is because they're great, is that we might be really interested in these weird birds and why they are the way they are and how they might have diversified over time. And so we could take all of the extant penguins, so there's like about 19, 20 or 18 or 19 species, depending on what you think about species delimitation, I guess. And we might reconstruct their phylogeny of just the living taxa, estimate their divergence times using maybe these calibration approaches if we have some fossil information that falls within the crown. And we get some picture of the diversification over time. What we see is that they've been diversifying since the early Miocene, and there's this many in the present, and maybe some of the more recently evolved groups might be have diversified in conjunction with something interesting in terms of climate or biogeography. So that might be cool, but if we think about penguins in the context of the bird phylogeny, what we do see is that they've diverged from their most recent common ancestor with their sister lineage, which is these procellular forms. So these are things like albatrosses and terns. And so they've diversified in the late Cretaceous. So if we think about this tree that was estimated using these calibration methods just on the extant taxa, there's actually quite a lot of things that happen between the split between flying birds and penguins. And if we're really interested in penguins and why penguins are the way they are, I think there's quite a lot of information that we're missing if we're not considering the evolution that happened along that stem lineage. And if we think about what makes penguins so interesting is they have this very, similar morphology because it's associated with their habitat and their life history. So penguins actually fly underwater. And so what we see in penguins and in the fossil record in their bone morphology is that they have this very distinctive shaped scapula. So flying birds have this very narrow blade-like scapula, and penguins have this broad paddle-shaped scapula. And that's because all of the muscles that attach to the scapula actually have to move water through space, right? Or they have to move through water instead of through air. And so that, because of that friction, that ends up becoming a very challenging thing for a penguin, and so they need this more sort of leverage to do that. So we see these like amazing sort of character evolutions in penguins that we don't see in other birds, and this is, you know, really critical, I think, to their life history and biology. Additionally, we see that penguins are really terrible at walking on land, and that's because they have this short, square tarsometatarsus. There's so many videos on YouTube called Penguin Fail of them just tripping, so it's really great. But so they don't walk very well on land because, so all birds have this fused tarsometatarsus where it's the fusing of the ankle and foot bones, but penguins have these really short square ones, and we see this also in the fossil record. And so there's interesting aspects of their bone morphology that make them, you know, fascinating to study in a macroevolutionary context. And another thing about penguins is they have solid, flattened wing bones and solid bones overall, so this makes them great at fossilizing, but also makes them interesting to compare to other birds when you think about the, you know, how flying might, you know, drive the biomechanics of the bone morphology as well. So given that we see all these things in living penguins, we also see some of this in the fossil record. So we have, you know, a few penguin fossils that fall within the crown group of penguins, and so this is one that falls here in the phylogeny. And these also have many aspects that we see in the bone morphology that we see in living penguins. But we also see penguins that go back quite a long time before we've estimated the age of the most recent common ancestor of penguins. And here we see, you know, slightly different aspects of their morphology. They often are quite a lot bigger. They have these like very spear-like bills, and some of these stem fossil penguins have scapula and their tailbones, the pygostyle, that's more similar to non-penguins. So this is one that was from approximately 27 million years ago. And the oldest penguin still has this short, square tarsometatarsus, but has a relatively intermediate wing morphology. And so what Dan says is that this penguin, so this is the oldest fossil penguin, and that this penguin probably was not great at walking on land, but also wasn't that great at swimming underwater. So, you know, given that we've seen this history in the fossil record, but also an immense amount of diversity in fossil penguins overall, you know, accounting for this part of their diversification is pretty critical to understanding the diversification and biology of the living penguins. And so these are just reconstructions by artists of fossil penguins, so they fall on these, and then these three are living, or these two are living penguins, and this one's a crown fossil. And so what we did is we did this joint estimation of the phylogeny with the fossils, and so we had all 19 living species and 36 of these fossil species. Then we had 202 morphological characters and over 8,000 DNA sites. And so this is work with Sasha Gavroshkina and several others, and this was actually implemented, or we did all this work in BEAST. And so we co-estimated the phylogeny of this group with these data. And so what I'm showing you here is some representation of the resolution of the support posterior probability. So it's above 75 that I colored in black, but you know, most of these were in the 90s. But then, you know, several of these fossils in this summary tree fall out as sampled ancestors, but this is the relationships that we have in this sampled ancestor maximum clade credibility tree. And so we see a lot of the fossil penguins falling out on the stem, just like we've inferred previously by, you know, what Dan has done in his papers with parsimony and other approaches. So that's pretty consistent with previous studies. But here's where I'll take a minute to also comment on the fact that, you know, one of the issues with trees that we're summarizing from MCMC, it's really difficult to represent our uncertainty in relationships in a visual way. So we might look at this tree and we might say, oh, wow, well, you know, it turns out that this fossil's you know, related to this group and is a sampled ancestor. But unfortunately, like, you know, presenting this in a summary tree is really challenging. So we still do need better ways of saying, like, you know, what our uncertainty or where we can't make, you know, overconfident statements about relationships or times or anything when we're representing it in a summary on a tree. And so that's one word of caution, I think, to take when looking at these things and applying these methods. But, you know, given that we can try and summarize our data and uncertainty across, you know, varying different ways, then I think we can get better at approaching understanding that. But so, yeah, so these are the relationships from the total evidence analysis. And one interesting thing, and this made Dan happy, was that we actually estimated a much younger age for the most recent common ancestor of living penguins than had been previously presented in the literature. So previous studies, so here are two previous studies. This Baker et al. Study used penalized likelihood, and some cross-validation approaches for getting uncertainty. And this other paper used a Bayesian approach with these calibration density methods. And so when we compare our estimate to those, we get a much younger age for the most recent common ancestor. So previous studies had estimated that to be about 20 million, and ours was estimated to be about 12 million. And this is because what our understanding is the diversity and diversification of the stem lineages is providing information as well as the morphological data about the evolution of the characters and the diversification process in this tree that leads to informing that younger estimate. And so what's interesting, though, is when we just take this group here and exclude all these stem fossils, and don't account for the diversity on this lineage, we end up estimating a much younger age for the most recent common, or much older age for the most recent common ancestor of living penguins. So even in using this fossilized birth-death process, not accounting for the stem diversity gives us a biased estimate and different estimate of the age of that node. And so what this indicates is ignoring some of the data and, you know, not accounting for these observations of lineages back along the stem is also going to potentially, you know, like ignore aspects of the process and mislead methods that are trying to estimate these processes. Yeah.
So you get these two different results. How do you know which one's better?
Well, we're going with the one that uses more data. So if you throw out some of your data and get a different answer, but you still think those data are real data, then, you know, that's the one that.
As long as the model that you're using to integrate that data.
Yeah.
Is appropriate.
Right. Sure. I mean, these are all the things that we have to sort of stand by, right? It's like, you know, we're saying that this is the posterior probability of the age of most recent common ancestor of living penguins, conditional on my model and my data. So that's what a posterior probability is. It's conditional on the rest of the model and the data. And that I feel pretty comfortable with. And absolutely, if we discover a fossil that you know happens to be a crown fossil, but also happens to be, you know, 40 million years old, then that would probably change a lot of this too. But there's a lot of filtering when it comes to the fossil record, right? There's only so much we can recover. The biggest sort of missing data issue, I think, that's out there is fossil data. And so, you know, you might also think too, if you say work on molecular things, if you're only sampling one gene and that gives you one particular answer, and then you sample another gene and you get a different answer when you combine them, how does that change your prior belief in the combination of those data. So that's sort of would be the approach that you would consider. What we wanted to demonstrate here was that, ignoring that this is sensitive to removing removing data as it should be, right? Like, if you remove data, your estimate should be sensitive to the removal of those data, either in the amount of uncertainty or, even in the mean estimate overall. Yeah.
That's quite a striking difference with the things, right, between the black and the red codes. But I imagine like the richer the fossil record and the more molecular data you have in the crown, the smaller the difference, right? Must be
So
as you know things happening along the stem.
I think even if we had full genomes for all of these, if we didn't account for this, we might still get something similar to that. The thing, because of this, the compound parameter, there's really only so far you can reduce the precision or reduce the uncertainty in node age estimates because of this, joint estimation of those two parameters. So with more molecular data, you'll only shrink that so far. If we had more and more and more and more fossil data in here that was also consistent with this, then yeah, that would work to do that too. But what this is overall doing is this joint estimate, and this precision, this is much more precise because we also have more data. So as for anything, for something to be sort of statistically, to make sense statistically, as you add more data, you should increase your precision or reduce your uncertainty, right? And so, we've included more data, and therefore we get a more precise estimate too.
Given that for the fossil record you only have morphological data,
Mm-hmm.
right? So does that mean that it's really the morphological data that's pushing the time more recent?
So,
Dedicated sort of pushing in the other direction as the morphological
So
data starts driving the time more?
there's nothing pushing without these, there's nothing pushing this in any direction. It's just sort of, it could be, well, you might have some of these fossils that might keep it from going in certain directions, right? It's the combination of morphological data and the occurrence times are data. So we're treating them as observations here. So the combination of those two things are really what's driving, or are part of what's driving this. And our priors on the speciation and extinction rates and stuff like that. But so it's both of those things, because yes, our relationships here are mostly informed by the morphology and the times, right? You obviously, given that this time is here, it's unlikely for these two taxa to be sister, but also their morphology might indicate that they're not sister. But so that's why you have these things sort of happening in conjunction.
And how about the parameter estimates within the crown? Like how, I imagine like the node ages within the crown are still also affected?
Yeah, I mean they should be, right? Because everything's going to be, well, they may be, and it really depends on some of these things, because given that you have fossils here, might still not have a huge, changing this might not have a huge impact on some parts of the tree, but I actually don't remember, we did look at some of that, but I don't remember what the details were. The one thing is that we did get a different estimates of ages in this group that gave some indication of sort of a different timing of the colonization of Antarctica or something. But those are penguin details that I don't remember so well. But yeah, so in some cases they may be influenced, but maybe in the places where there a strong support for fossils being part of that tree, then they won't have a strong influence.
Right.
So are there any other questions about penguins? So this is a study that we published this year. Actually, it's 2017. And so it's kind of really interesting to think about, these types of processes and how they work on a fossil record like penguins. And one of the things that becomes important when you're looking at a group with a rich fossil record like this group, and this is actually, or the bears, I guess, in this example, is that for many groups, these fossil ranges don't actually represent uncertainty in the time for a single specimen. They represent these stratigraphic ranges. So this is, the first and last occurrence of multiple specimens that have been identified as a single lineage or species over time, right? And so given that we have this other concept of what the data actually are, it then becomes really important to think about how we might model that kind of data, as opposed to one that just simply assumes you have a point estimate for every specimen that you've observed. And so given that we have this different kind of concept of what the paleo data are actually represented as, it becomes important to model them as stratigraphic ranges. So we have just posted this archive preprint on this model for stratigraphic range data. So on some ways we can also, in paleontological literature, often the stratigraphic range data are not presented in a phylogeny. That we might have like here, 100 lineages of bivalves and each of their first and last occurrences. And what these approaches are seeking to do is look at how diversification might change, say, over the time of that group, because of varying things, right? And so given that you have the appearance of a fossil in one part of the fossil record and the disappearance in another, then you have this varying ways in which they are sampled. So I'll just talk a little bit about this biogeographic stuff, and then we have a coffee break. But so there's this other issue where many people actually have no fossils. And so if you work on something like amphibians, which actually have a really poor fossil record, you might be wondering, like, how then do I estimate divergence times? And so this is work by Michael Landis, and he actually, if you go to this link, which I'll give you the slides later, he has a nice little YouTube video of his talk that he gave on this. And here's the concept of where we might actually have biogeographical information for some species in our group, and we have some model of how that biogeography allowed organisms to go between areas in the biogeographic history. And so for this group, let's say they're found in 2 areas, and we know at some point in time those areas were contiguous and species could go between A and B, and then after some point in time A and B were no longer accessible, so you couldn't go from one to the other. So given that we might have this type of idea of the history, we can then sort of assign probability to dispersal events on our tree. So if this is our tree and these are our data, and this is the time at which the event or the dispersal probability would change, if the dispersal happened after this point, then we know that to go from A to B was, this whole reconstruction is less probable. Because in order to have a split here, we would then have to have an ancestor in this lineage dispersed to area B. If we move this node so that the dispersal happened when it was actually a probable event, then we can assign a higher probability to that. So this is this concept of having epochs over time where the dispersal to and assignment of lineages to different areas has a different probability. And given that it has different probability, then that will actually have an effect on the ages estimated for that node. We can also think about the reverse event where we have 2 areas merging. So here we would have a rare event where we now have 4 taxa, and we have a Sicilian now found in area A. And if these 2 are found in B, when did this dispersal from A to B happen in the ancestral lineage to the leopard frog and the red-eyed tree frog? And so if our divergence time is here, then we have this dispersal event happening here, and that should be a less probable event. Whereas if we have our divergence happening at this point, and the dispersal event becomes higher at the point in the epoch where the areas are connected to each other. So if we think about this in this constant, if we have only a single epoch where you have constant model of area ranges and dispersal probabilities, then you can say, we can think like, oh, we can move our nodes any point in this tree and they all have this basically equal probability. But then if we overlay these epochs where now we have maybe some higher connectivity between dispersal, then we can think about how our model might be able to understand how the probability of a node being in a certain time might change over those epochs. And so this we can actually bring into this framework in a Bayesian sense, where we can allow this to actually impose a structure in time for our divergence time estimates. And so here's just these broken down into two things where you actually have these models are, essentially you have a single rate or a single sort of model on your on a branch, versus when you have an epoch, then you break this into two different models for that branch. So then for Michael's just published this in a paper this year for essentially talking about how you can date phylogenies without fossils. And so the input for this would be molecular sequence data and biogeographic ranges. But then you also have to have this essentially an empirical paleogeographic model that describes the connectivity over time. So this is sort of underscores another point when, if you're interested in these types of approaches, really understanding the geography of the history of the area that you're interested in becomes important. And so much like for working with paleo data, where it becomes critical to collaborate with a paleontologist, it then becomes important to also consider collaborating with geographers in this case too. And so what Michael did is he developed a paleogeographic model that describes connectivity over time in 25 areas and 26 time slices. So if we think about continental drift, and as these things connect over time, you would see that you have areas of weak connectivity and areas of strong connectivity where your organism might be able to disperse as the continents shift over time. And given that we have this information, then we can construct this as our paleogeographic model. And so what he did is he used this and applied this to the turtles, which actually have a pretty old history. And he compared this also to estimates from the turtle fossil record. So turtles have a great fossil record, and you might not necessarily need to use just biogeography, but when you have both, you actually get really good estimates for divergence times. And so the other aspect of this is that what you're essentially doing is integrating a biogeographic framework for understanding your macroevolution in your group with co-estimating divergence times and potentially tree topology and other things. And so then accounting for uncertainty in that part of your analysis while co-estimating the geography. And so you get things like ancestral area reconstructions as part of this analysis as well. So that's just one other aspect that will become relevant tomorrow when people talk about biogeography, because that is part of tomorrow, right? But this is, and they might not discuss the dating part of it, but you can actually use these data to date your phylogenies.